So we're going to take questions from the audience, but what we did was we actually took questions online on social media over the Thanksgiving weekend, which is what we generally do every year because it gives families time to think about the topics of the discussion and what their questions are and have a chance to kind of write them privately to me, because a lot of families are just not comfortable standing up and asking you guys. You're very, very intimidating. If you couldn't tell, I'm very intimidated. And so I am going to speak on their behalf and ask you questions, and then we will open it up to the room, certainly, so anyone who does want to ask a question. We don't want you to feel intimidated, and we want you to be able to come up here and ask. So I'm just going to kind of randomly choose some questions for each of you, and I also thought of just a couple that I didn't know what was going to be the content of your talk until afterwards, so now I have some questions as well. So I'm going to start with Craig Martin. Where is he? He's here, right? Is he here? He's in the room? Oh, there he is. Yeah, get up here. Of course, I'm not going to start with Craig Martin. I will start with Dr. Dindo, who I know is here, because you're easy. There are a couple questions for you, actually. One question was, and this was for you and actually Dr. Kyung as well, from a genetics standpoint. Can you explain your concerns about how you are testing or considering overexpression of UBE3A in paternal activation for the UPD and ICD genotype when it comes to paternal replacement or a paternal activation of UBE3A compared to the option, if they had both, of maternal replacement of UBE3A? So for UPD and ICD, can you first describe your concerns about paternal activation for that genotype? And then, knowing that we may have in the near future a maternal replacement option, do you feel genotypically that that is a better option for that population? I'll start with Scott.
So the question is having twofold expression of UB3 if we completely turned it back on. So there is evidence that overexpression of UB3A can or is associated with cognitive deficits. You have Dup15q syndrome, which is it's a large duplication, and oftentimes there are multiple copies of not just UB3A but several genes, and it's not entirely clear what the contribution of those other genes are to, like, say, that condition. In cases of Prader-Willi syndrome, you'll have like a UPD Prader-Willi syndrome where there are twofold expression of UB3A because of both alleles being expressed. So there is evidence that that's the case, that too much UB3A is bad. What we've seen in with ASOs and even in some transgenic mice is that, especially with ASOs, for example, it's hard to get a twofold level of expression. I mean, you can get it if you completely turn the antisense transcript off. But most oftentimes what you see is some gradient within that range. My personal opinion is that I think it could be, it's something to be aware of, but I think it's something that can be managed. I mean, if we're talking about an ASO, we know that there's a dose-dependent relationship with the amount of ASO we give and the amount of reactivation we get. So knowing that, I think there'd be a way to control how much is potentially being increased. But the good part of that is that it means that it requires a much lower dose, or it may require a lower dose. And so I think that's a good position to be in.
I'm going to follow up with that, and can you turn on this mic as well? I'm going to follow up with that with one more question to you, because I think the next question that I know our UPD and ICD community is waiting for me to ask, so I'm just going to ask it for you, Lauren, is that if you, obviously depending on the target of an antisense or CRISPR or whatever the technology is that you're activating the paternal, are you concerned about overexpression of, or double knockdown in either way, I guess depending on where it is, of the SNO RNAs? The impact of paternal knockdown biallelically and the impact that that would have on the SNO RNAs.
So using an approach where you do actually repress them?
Mm-hmm. Like if you are repressing them, but marginally, so it's not a big deal if it's on one allele, but now these kids are used to having maybe potentially two alleles expressing SNO RNAs. What happens if you knock them both down? Is there any concern that you see there as a problem?
To the best of my knowledge, and maybe some others can chime in, there's no known problem with having too much of the snorinase.
There you go, Lauren. Told ya.
To my knowledge. From a genetic standpoint, it's clear that the SNORD116 in that region, the Prader-Willi syndrome critical region, something that's made in that region is what underlies the Prader-Willi syndrome. It's less clear what the snorinase downstream do, and so there's evidence in the literature that, at least in mouse models, that deleting the SNORD115, for example, doesn't have that phenotype. It seems likely that they would, because they're genes and they most likely have some function. Another question too is the sort of developmental timing of those genes. So if you take, for instance, a treatment in a child or an individual, it could be that this happens later in life. It could be that the function of those genes is important during brain development in utero. And so having them reduced is no longer an issue. But just speculating here, I think it'd be best to kind of look at these things in a very specific context with their relationship to these treatments and see what happens.
And I think that's what we're trying to do with Albert's work, is to be able to look at it in a 3D model or a 2D model to understand the phenotype of that model and see how that phenotype changes for better or for worse once we double activate or double inhibit, essentially, the antisense and double activate the UBE3A. So I think that was the question directed at you, but I think Scott just helped you answer it. And Dr. Jiang, I also, if you have anything to add differently as a clinician and a geneticist for the UPD and ICD community, as the antisense knockdown approaches seem to be approaching a bit faster than the maternal replacement approaches, and these communities and these families want to be included in these trials, and we want to do everything we can to de-risk whatever it is so we can ensure that they have the same opportunity that every other genotype has. Do you have anything else you can add there?
I would add, to me, I conclude we just don't have enough knowledge to conclude anything now.
So how do we gain that knowledge so we can start figuring it out in humans so that our pharmaceutical partners are comfortable including these kiddos in their trials?
For a simple reason, we think that antisense come from somewhere from the imprinting center that's megabase away. We consider it's 1 megabase transcript. It's a continuous, that's by presumption. So therefore, you use the antisense oligo in one location, are you going to be disrupt entire transcript? Just destroy that portion somehow critical to reactivate UBE3 gene. Then that antisense can skip alternative splice and jump into next exon. We just have a little knowledge to me. That's what I think the iPSC organelle can help us, particularly UPD. I think that's what I think the vision we put on, create a UPD iPSC cell line, that testing that, get more knowledge. But on the other hand, you and me kind of talking very early, for the community, the parents of UPD imprinting mutation, I think the hope is great.
Yeah, so they have potentially more optimism, yet they're feeling pessimistic because people aren't including them in trials. And so we need to close that gap for them. And so I think ultimately we're doing what we can from a preclinical perspective, but I think human data is really going to drive understanding. They may be the best responders.
Yeah. Adding on for the parents with imprinting mutation, oftentimes you got a report, actually you don't know which type it is. So I met so many families have one perception where their kid's mutation is. So that's imprinting mutation as a 2 type. One is you clearly have a deletion in the imprinting center. One that you don't have it. That's 2 separate group 2. So it's even the small, oftentimes there is no clinical testing besides the methylation tells, oh yeah, you are not on the UPD, but you cannot separate either imprinting mutation with the microdeletion in the imprinting center versus the possible transacting mutation outside of chromosome 15, actually acting on the imprinting center cause the imprinting mutation.
Right.
Can I add just one thought is that I think that this difference between a deletion and UPD might not be really the way to think about it. I think that it's really just about the total amount of UBE3A, right? And the UPD does have 2 copies that can get reactivated, but there's actually a lot that we still don't know even about the large deletion situation when you're adding an ASO. So the analogy I would think about is if you think about the brain as like a jar of jelly beans, and you had some magical thing where an ASO, you add an ASO solution to the jelly bean jar, and it can turn the jelly beans red, like UB3A, right?
We like red.
The key issue is that the way that we generally measure red is that we would add the ASO, jelly beans would turn red, and then we would mash up all of the jelly beans and then measure total how much red is there, right? When you mash up all the jelly beans, each jelly bean representing a cell, when you mash up all the cells together, you're averaging the UBE3A from all of the jelly beans, from all of the cells. You can get, let's just say 100, your UBE3A level is 100, just an arbitrary number, 100 units of red. That can come from one jelly bean out of all of the jelly beans being really, really, really red, and then all of the others being white, right? Or it can come from every single jelly bean on average having 100. So there's this heterogeneity that can be an issue, and that's actually something we don't really completely understand with large deletion either. And I think if you think about UPD and large deletion in that same way, then they're kind of, similar in that sense. That's one of the things I think these organoid models are really useful for, and animal models are really useful for, is because you can, with newer technologies, start to look at, how each single cell is behaving. So.
Yeah, well, that's why we very much look forward to your data when you start putting ASOs on these organoids of UPD and ICD. So next year, stay tuned. Okay, next question. This is going to go to Dr. Ziegler. How likely is it that the ORCA will be considered, this is a good question, as a primary endpoint in AS, being that it's caregiver reported, yet the most accurate measure for communication in our kids and the most meaningful to most families? And this may be something that Jennifer has to support from a regulatory perspective.
Yeah, so I think one of the things about determination whether a measure is fit for purpose is it depends on the specific drug program. First of all, it depends on the data collection schedule, and it really is ultimately a judgment call based on all of the documentation and the data that's available to support a measure. So I think starting from a place of documenting really rigorous qualitative and quantitative support for measure development puts us at a really good place. I think the answer, it depends, is never a very satisfying answer, but it really depends on the mechanism of action of the drug, how quick we think it's going to work, is communication ability really the primary target? And then it comes down to all of that documentation, and can we build that rationale that this is the best way? And often I would say that caregiver report and clinician reported data really complements each other. It tells us different things. And so that might be a way to get a full picture of what's happening within a clinical trial. And Jennifer.
Jennifer, do you want to add anything?
I would also say it depends.
She's my litmus test. I have all the optimism, and then she's my litmus test, and she settles me down.
I think the work that's being done now is to get it in shape where it could be a primary endpoint, but it doesn't mean it will, right? What's going to happen is all of the trials are going to evaluate where they see a response in patients, and there may be another endpoint that does a better job of showing that. I think what's important is that we have multiple tools that could serve that purpose, and what's really great about the ORCA is that we know that it's meaningful because it's capturing an assessment of communication, which in the survey and in the disease concept model were ranked the highest by caregivers as a change they'd want to see.
Great, thank you. This question is actually for Dr. Zhang and Dr. Zhu about your presentation. The question is, how long do you expect this new technology could take to get into clinical trials for children living with Angelman syndrome? See, we get right to the point. I make them turn red.
Alice and I are definitely working very, very hard, that's for sure. We know the potential and the promise. So when you have a potential promise, we will definitely working very hard. I just cannot give you the timeline. Maybe 3 months right now, I can give you very much definitive timeline if we get that $24 million.
$24 million. Are you guys ready tomorrow night to paddle race? $24 million.
But on the other hand, I would probably say, regardless, we would continue working on it. You see the promise there, then we feel that's excited, that's direction. We would continue work on regardless. So hoping 6 months, 1 year from today, then we say, oh yeah, we are in the next phase.
Great. So we're in preclinical. We're working toward IND enabling.
Yeah.
And we have human candidates. So therefore, or close potentially, we're probably looking at a few years, but closer than early discovery work would say. Would you support that?
Yeah.
Okay, fair. Okay, next question is for Dr. Pitluck and Dr. Jarvis. If a patient is in a trial and it is not a one and done, and then a drug goes to market and gets approved, does that patient then need to pay for that drug, or would they be provided long-term by the company that ran the trial? That came from the community.
Did you say Dr. Pitluck? I just got
Oh,
promoted.
you got promoted to doctor.
That's great.
You command the stage as such.
I love it. My parents would be very happy. So the short answer is that after regulatory approval, payment for the drug, the pharmaceutical companies, who's ever sponsoring the trials and the drugs, are going to try to work with the insurance companies to get payment, particularly in rare diseases where you have high cost products. Getting payment out of pocket is very challenging. So the goal, not only in the US, but outside the US, is to make sure that the payers, both public, Medicare, Medicaid, mostly for the Angelman syndrome children, private payers, whether it's Anthem, Aetna, Blue Cross, United, that they are prepared to pay for the trials after the drug gets regulatory approval. Remember, for gene therapies, though, in the one and done approach, when you have the drug in a clinical trial, there's nothing to take afterwards. So from the gene therapy perspective, once you get it in the trial, you don't have to worry about reimbursement after regulatory approval. Where the reimbursement becomes an issue for regulatory approval is for children who did not have it in the clinical trials. And the goal is what we talked about earlier, is making sure that payers are ready to reimburse the drug once regulatory approval is achieved. And that is what FAST is working on doing. Studies like Medicus is working on the caregiver burden survey, so we could have all the parts in place to have reimbursement upon regulatory approval.
Okay, that's a great answer. All right, why don't we open it up to the room? If I have many more, but I want to make sure people that came get all their questions answered. So I knew that first hand that was going to go, it was right there. You go ahead, Lauren. Can we have a microphone back there? There it is.
Hi, this question is for John Jarvis. I wanted to make sure the Caregiver Impact Survey asks about how much cost families and insurance companies are what we think we're paying to take care of our kids with AS over and above what it costs a typically developing person to live. Can you help us understand why that's important?
Thank you, Lauren. That's a great question. The question, why is this data from this Caregiver Impact Survey, which we've asked you all to take, thank you again. Why is it important? Let's take a Blue Cross Blue Shield. A new payer is looking to cover a drug that's been approved by the FDA for Angelman. One of the first things that they're going to be thinking about is what is the total cost or what's the total burden of this condition right now, even without treatment being available. The industry term is burden of illness. For some of these other rare genetic conditions, we can look to DMD, SMA, research from EveryLife that's come out over the last couple of years. We say that a huge portion of these costs are borne by the caregivers. So whether that's in the form of lost work time whether that's in the form of out-of-pocket household costs, whether that's in the form of just informal time spent caregiving, makes up a huge portion of these costs. The goal of a survey like this is to make sure that those costs are not forgotten about, that they're front and center, that they are additive to the other costs for medical, hospitalizations, doctor's visits, et cetera. So that's the purpose here. And just quickly plug the survey again. Thank you all for the great questions. We've enjoyed the discussions a lot. So thanks for filling out that survey. Thanks, Lauren.
Alison, can I add on something else?
Of course.
Yeah, so this is a little bit deviated the original purpose, but I think when we think about newborn screening, and the one thing we will be asked, the cost. And sometimes it's very difficult to calculate. So you need clinical data, and I think this survey can use one stone, two birds type thing, because if we are ready for the nomination going put forward, this data will be helpful because you calculate the cost, not screening, right? So if you're not screening and the parents go through the, not only life you save, but also the medical bill, because clinical diagnosis odyssey. And the caregiver have to lose a job, we heard today, right? And each time you go to clinical, the appointment, and your kids need to go to ER. So I think this is survey I want to plug in. I think is terribly important for other purpose.
I'm going to ask a question about newborn screen actually for Dr. Kucera. You have a microphone back there? So this is for you. How long do you feel it will be before AS actually gets on the RUSP for a newborn screening in a state? And then how long does it take to get on each state generally? That's the first question. There's two.
Yeah, that's also a hard question to answer, right? The timing. And May is probably more qualified from her longer experience in newborn screening about how long it takes. So we are at the beginning, right? We're developing and validating assays in a research kind of environment. And so it takes a couple of years at least to run a pilot study that generates enough data. So we need to show that it actually would work in a large throughput and kind of environment. And we need to identify newborns through newborn screening to demonstrate how the entire process would work. So a few years of piloting this in a consented kind of study, perhaps. And then a full-scale pilot is usually helpful as well. And then there's a lot of evidence that needs to be provided to the RUSP committee for evaluation that includes not just the screening, but treatments and what kind of benefit there is to public health. We are involved with the nomination for Duchenne, which is currently in the process, and it is a lot of evidence. It's typically or frequently the foundations that pull together the applications. And there are always a lot of questions from the committee in the process of evaluating how appropriate it might be. And then if all goes well, then a condition gets put on the RUSP, and each state has a different policy about how to implement it. So for example, in North Carolina, when a condition is added to the RUSP, North Carolina has 3 years to implement it. And it is a big deal to implement it, so they do take that long, if not longer, to actually do that. So it is certainly the right time to get going on this process because it is extensive work and a justification that this is appropriate for public health newborn screening.
Yeah, it's harrowing, actually. It feels harrowing how much work it is, but I'm glad we're starting now because I expect approvals in the next 18 months to 2 years, so work hard out there. Okay, any other questions from the audience? Sorry, I just can't see. There's a hand. Oh,
question for Dr. Kucera and Dr. Baker. Has the newborn screening changed our understanding of the prevalence of any of the diseases that you screen for? And number two, how much does a newborn screening cost?
So the question was, Dr. Baker, maybe you could take this. The question was, has the newborn screening changed your understanding of the prevalence of the disorder, and how much will a newborn screening cost?
Okay, this is not a simple answer. First, I think this morning I was made a comments. Usually we see is the newborn screening really change the prevalence the things that we know ahead time, because it comes from the clinical expectation. And for the SMA in our state, I see a little bit higher than we expected, but also some state experience a little bit lower. So people start thinking why. It's maybe some prenatal carrier screening kind of influence because they make a reproductive decision. So I think we need a time to see on that.
What does it cost?
Okay, newborn screening, I think when it costs, we need to think about not just the testing. I can tell you the asset today I present to you will not cost very much. Think about it, we already have DNA. So we do need to do the methylation piece, bisulfur. And the testing, I was thinking around $15 for testing. But even think of this cost, so you have to think about the follow-up. The follow-up, you're thinking that potentially, if you have so many false positive, they will have the pressure on the health system. So I'm adamant, say, let's try very, very hard, minimize the screening false negative. And also, to me, on the Angelman syndrome, my hope is knowing that actually the downstream actually help save the health system dollar. I don't have numbers, it's why we have John and other people, we work together, get them, but I think it's terribly important.
Thank you. Any other questions from the audience? Oh, over here, David Kersey.
Thank you all for your talks. This question is for Jennifer. Could multiple studies, if they were undertaken at the same time, share a placebo group? And if so, would that be something that FAST would coordinate, or maybe would that be a role for the Translational Research Centers of Excellence?
There are study designs under what's called like a master protocol, which is sort of a big overarching protocol where you could study different types of drugs and use a shared placebo group. This happens in cancer research, and I think Duchenne muscular dystrophy as a rare disease community worked on a master protocol, and it basically allows you to follow sort of the same set of assessments, and then you don't have to enroll as many patients on placebo, so it's a good question. That works really the best when you have similar types of drugs, like so not probably a multiple dose drug and a gene therapy. But it's definitely something we could consider as a group, absolutely.
Liz, get on that. Okay, any other questions from the audience? I have a bunch more. There's something back there? Yeah? So you're asking about, the question is really about prenatal screening, and if Angelman syndrome is going to be or is on prenatal screening, amnio or CVS or the non-invasive testing. And I don't know if anybody here wants to answer that. I can certainly try to answer that. Are you guys good with the prenatal as well?
Yeah, I can take a stab at it. We are not involved in prenatal screening, so that's different. And I'm not sure if I heard the start of the question correctly, but I think you were asking about
what else is on newborn screening? What else would they test for on newborn screening?
Yeah, so the newborn screening panels currently have a number of conditions already. And the process of nominating new conditions is one by one, unfortunately. It is actually one of the issues in the field. There are all these new therapies that are coming out, and the process is quite slow because it's one condition at a time. So right now there isn't a mechanism that I know of that could bring more conditions at the same time. But for example, sequencing is one of the opportunities, the technology that would make a lot of sense if you can screen for a lot of conditions at the same time. It is expensive, but if you start screening for hundreds of conditions, then it becomes a lot less expensive per condition. So there are several different groups that are piloting sequencing-based studies for rare conditions. So that is on the horizon as well, but it is very different from what the current system is, and it would really require a major change in newborn screening.
I may add on for the prenatal. It really depends on technology. Right now, some of you know, we do the NIPT testing for trisomy 21, 18, 13, or trisomy X, because that's almost like a universal screening for all pregnant women. Actually, interestingly, my mentor, Dr. Baudet, and he identified the UBE3A gene, and he dedicated his time to develop a new assay, can do microarray for in the mother cell for the prenatal. If that work out, I think he's still working on the technology side. It's if it work it out, then you say, oh yeah, every woman, if you want to get the testing before early pregnant, that's become very, very real possibility. You can do the prenatal testing very reliably and also pretty kind of cost-wise, pretty acceptable for the community. And then you can like the current, almost most of women over 35 get NIPT testing.
Yeah.
If I may, I'm adding on some comments on newborn screening. So you saw my slides. I was a representative that have multiple disorders, right? So in newborn screening setting, you need to remember not every single one is a genetic condition. So use gene panel, use a whole genome. That's not only answer. So you have to be careful with that, because the disorders on the panel, congenital hyperthyroidism, you cannot do genetics. And that's one thing. Second thing is the timing. I mentioned that. So fortunately, I think at Angelman, we do have a time to react. CF, we do have a time to react, but the organic aciduria, the amino acid, urea-cycle disorder, sometimes I feel newborn screening a little bit too late. So I just want to give you some simple example. So to me, in the newborn screening, feature hold is, we heard so many times, not a one fit all, right? We heard that. I think in newborn screening, I see that the function analysis and the genetic components have to be complementary. So I think indeed, to me, I think importance and Kate mentioned that it's a study, and actually our program involved that too. So we need a lot to learn. So if you ask me today, I would say no one fit all. So we need really is the outcome driven, the utility driven, what do we want to achieve? Then we go back, what is the best method to take us to there? So that's my 2 cents.
Thank you. So, oh, you have a question? Dr. Weiss, what was your question?
Uptake or distribution of your
or will it decrease like other techniques when you move to that larger element?
Let me just repeat the question real quick because he didn't have a microphone for the virtual attendees. So he's asking, how is the biodistribution of the technology with the CRISPR RNP from the Yale group compared to others that we've heard about, ASOs or AAV gene therapies, where the biodistribution seems much less compared to what they reported? And is there an expectation that will change when we get to the larger brains, like in non-human primates and humans?
Yeah, so for the first answer, it's for the cell already edited, that will not disappear. We believe, because it's a DNA, it should continue lifetime if that deletion or editing already happened. The question is, we kind of still will continue evaluating. For example, this is like a P1, a day 1, day 2 in the mice. We need to ask our second question, if we deliver mice like P21 day, postnatal 21 day, how good the distribution is. We don't have that, we are working on that data. Now come to the human, we don't know for sure right now. So we're gonna try to using one kind of organelle to deliver to the organelle. This technology being developed is really amazingly efficient. You just incubate these things in the culture and the petri dish, they just go into the cell very effectively. So now probably the best we can get from the human, how this distribution with the human brain. Of course, after the next to the human will be monkey. If we can deliver to the monkey, see how good distribution. So far from the P1, P2 delivery in the mouse brain, indeed, I did not show you data for the whole brain imaging. Pretty good, but of course, it's always has variability. I show you probably the 75%, that's the image we get. It's really amazing that different brain region may have a different variability, or we have not figured out yet. So the cell type of specificity, again, the cell type of specificity, as we know the neuron is pretty efficient. And we also check in the astrocyte or other, I just don't have time to show all the data. So it's remain to be seen kind of detail for sure. I hope next time in the meeting or other meeting I can show more for that detail. But it's a very good question. So.
All right, well, I'm gonna end with one final question. So our keynote who started the morning, I'm gonna have you end the afternoon, if you don't mind. So I think that you knew a lot about FAST just from our relationship and from you hearing talks that we've given and things that you've been following from the FAST story and the Angelman community, more importantly. After today and all of the talks that were there yesterday and what you see that's happening tomorrow, I'd just love for you to make a comment to our community, to the Angelman families who have supported everything that you've seen today, which I think is probably has a different color or feel to what you thought, to what there actually is, and just how that compares to other diseases like Angelman of similar prevalence, and where we are, and your optimism for that, or pessimism for that matter.
Well, I think in many ways it's been good to see at a more granular level what's going on, but I don't think it changes my overall perception of where you are. I think this community is fortunate in many respects, given the prevalence of the disease, to have so many areas of exploration about therapeutic development and so many other programs relevant to the prospects for drug development in Angelman. Many other communities don't have that, including some that are higher prevalence. So I think there's a lot to feel optimistic about. At the same time, I understand the impatience, right? We want to move forward faster. And even when we talk about things like screening and diagnoses and understanding the true prevalence, are there other ways to disrupt the system? And, I'm not one that likes to wait for government to move at our pace. So can we take advantage of sponsored testing programs and pool them together, for example, to leverage those in a way that could maybe support broader access to sequencing and other kinds of testing that might get us a little farther faster? Are there other ways that we could look to kind of innovate within what we're doing? And I think this community has a unique, in many ways, opportunities to do that, just given the brain power, me excluded, on this stage. There's tremendous potential. You've made great progress, but in my view, there's a lot of runway ahead. And so I hope you all feel pretty good about that. Lots more work to be done, but you're in a great place.
And that's why there's a roadmap, because we're not at the end of the road. There's a lot to do, but we've done a lot. And I think to end it there, I think who we need to thank for that are the people on this stage and the people that were on the stage yesterday, outside of our amazing community who does the fundraising to get us to where we are. But our researchers, our teams, that table I'm looking at specifically, who have been with us the whole time, and so many people around the world that are working really hard on the research side, and now on the clinical side, and on the development side, and our pharmaceutical partners. There's so much effort and work going into this for Angelman syndrome that I, as a parent, I said this last year, and here I'll cry, but I didn't choose to stand here, but you guys chose to sit here. Well, you two didn't, so you can cry too. But you chose to sit here because you care about these kids. I didn't choose to stand here, but I was forced to. That's a huge difference, and we thank you for making that choice, because you made optimism possible for this community who desperately needed it. So thank you. With that, let's go get a drink and go to the beach.